Small RNAs regulate many biological processes. It is therefore important to develop sensitive and unbiased methods to detect them. Our protocol provides a step forward towards this goal. Our protocol suffers from fewer bias issues than classical small RNA library preparation methods, and importantly, it allows a more sensitive detection of 2' O-methyl RNAs, such as plant microRNAs.
After extracting total RNA according to standard protocols, pre-run a 15% TBE urea gel for 15 minutes at 200 volts, and mix 5 to 20 micrograms of total RNA in a 5 to 15 microliter volume with an equal volume of formamide loading dye in a 200 microliter tube. After 5 minutes at 65 degrees Celsius in a thermocycler with a heated lid, immediately place the tubes on ice, and load the latter and sample on the gel with at least one lane between them. Then, run the gel at 200 volts until the bromophenol has migrated about 2/3 of the length of the gel. To elute the RNA, first, puncture the bottom of a nuclease-free 0.5 milliliter microcentrifuge tube with a 21 gauge needle, and place the punctured tube in a nuclease-free round-bottom 2 milliliter microcentrifuge tube. Next, incubate the gel at room temperature with nucleic acid gel stain in water for 10 to 15 minutes before viewing the gel on a transilluminator. Cut out the sample RNA between 17 and 29 nucleotide ladder bands, and transfer the gel piece into the punctured tube. Spin down the gel in a microcentrifuge at maximum speed for 2 minutes, and remove the 0.5 milliliter tube, which should now be